All right, the last one in this session before we go for a break is Boyan from University of California, Berkeley, in the field of computational biology. I'm not going to say your advisor's name. I'll let you say that because I will definitely get it wrong, so I'll let you do that in a second. And Boyan had a practicum at Oak Ridge National Labs in 2021. So I'll pass it over for the last talk of the session. Thank you.
Thank you. Hi, everyone. It's really great to be here. I think this might be the biggest audience I've ever spoken to, so that's pretty exciting. So I'm a mathematician working on computational problems in molecular biology. And in this talk, I'll discuss some ways in which I've applied mathematical methods to study protein structures, which I've found to carry some interesting geometry. And this project is concerned with a class of protein structures known as Leucine-Rich repeats. And this analysis has been enabled by recently developed computational methods for predicting three-dimensional structure of proteins. So actually, this figure here on the left is a 3D structure for a Leucine-Rich repeat domain. You may notice that this protein looks a lot like a slinky. In other words, a curved helix. And one of the mathematical problems that arises in our attempt to better understand these protein domains is the question of how to geometrically characterize a curved helix, also known as a solenoid, embedded in three-dimensional space. So before I discuss that, I'll give a brief primer on what proteins are and where they come from for those of you who aren't biologists. So it turns out that the core molecular mechanism occurring inside of every living cell is the transcription of genomic material encoded in the molecule DNA into a molecule called RNA. And that RNA is translated into a linear chain of molecules called amino acids, which are linked by peptide bonds. And when this amino acid chain is finished polymerizing, we call the resulting molecule a protein, which contains on the order of hundreds or even thousands of amino acids. And this process is extremely fundamental and pervasive in life and is happening many trillions of times every second inside of the cells in your own body. And proteins are very large, complex molecules which perform functions in a cell. And they are predominantly responsible for traits and phenotypes in organisms. And the biochemical interaction between the amino acids in this polymer caused the chain to fold into a three-dimensional shape. And this turns out that the shape of a protein is a major determinant in its resulting function because it influences how this protein interacts with other proteins and molecules in a cell. And this particular protein structure shown here produces a pigment. It's an enzyme which produces a pigment, and the molecule that it binds to is actually depicted there. It was solved using X-ray crystallography, and this enzyme catalyzes a reaction to produce the resulting pigment. And the phenotype corresponding to this is the color of, say, a flower in a plant. And one of the central goals in computational biology is to understand how the structure of a protein governs its function. And in the last couple years, a significant advance was made to this end. A deep learning model created by the company DeepMind achieved unprecedented accuracy in predicting protein structure. And this is a non-trivial task which scientists have been working on for decades. Typically, protein structures are done experimentally via microscopy or crystallography. And this new method has made protein structures more accessible than ever before by allowing us to bypass these expensive and labor-intensive laboratory methods. In general, high-performance computing is critical throughout computational biology because many algorithms ranging from building phylogenetic trees to aligning DNA sequences are very computationally intensive. And this particular task of predicting protein structures is no exception. There are a number of procedures in this architecture of AlphaFold which require significant computing power. The first step being aligning to hundreds of millions of protein sequences, and followed by a couple steps that involve the application of neural networks. So for my project, I needed to run AlphaFold on many hundreds of protein structures, and the computing resources available at LBL were essential in this analysis. So let me give you some context about how I got into all this. My lab at Berkeley studies immune systems in plants, and a significant component of the plant immune system is mediated by these proteins called NOD-like receptors, which I'll refer to by the acronym NLR. So NLRs are responsible for binding to proteins called effectors, which are secreted by a pathogen. This binding is the first step in triggering a signaling cascade leading to an immune response. You can think of these NLRs as being a kind of antibody, which is designed to recognize molecular signatures of an infection, which under this analogy corresponds to an antigen. The resulting phenotype is known as hypersensitive response, which you can see depicted on the right here. This response is designed to control an infection. It's basically a kind of local cell death. In this illustration, there are these little colored shapes. These are called protein domains. You can think of them as being building blocks of proteins which are higher level than amino acids. So they might contain a couple hundred or even more amino acids within them. And they're sort of, they have specific functions within the protein. I might describe them as functional subunits. So the domain that I'm particularly interested in is the leucine-rich repeat, because this domain is the specific portion of the NLR protein which is responsible for binding to pathogen effectors. So I'll refer to these as LRRs. So many plants possess hundreds of NLRs, which can identify different types of pathogens. But even in the most well-studied organisms, we've only been able to characterize a small percentage of NLRs. So it's therefore an important and challenging task in plant genomics to annotate these proteins and characterize their binding specificity. And this is knowledge that is very applicable. We can use it to engineer crops to become more resistant to diseases. So here's another picture of what an LRR domain looks like. It's the same kind of slinky-like object that I showed in the title slide. And this is a 3D structure of an LRR domain that is bound to a pathogen effector. The LRR domain was taken from a model plant organism called Arabidopsis, and its target effector is from a fungal-like disease known as downy mildew. And as I had alluded to before, the binding specificity of this receptor is predominantly dictated by its three-dimensional structure. If the geometry of these proteins don't match, then this binding will fail to take place. And there's an ongoing ancient evolutionary arms race between these two organisms where the pathogen effector is continually mutating to avoid detection. And in response to this, the LRR is mutating in order to ensure that the binding takes place. So note that the structure actually only includes the receptor domain. It doesn't include the rest of the protein. And that just kind of demonstrates the fact that only this domain is necessary to achieve the binding. The rest of the protein serves other functions in the receptor. So this begs the question, how do we systematically identify the LRR domain within an immune receptor? So the task of, in general, identifying these functional subregions of a protein is called domain annotation. In other words, you're given the whole protein sequence, such as the one shown on the left here, and the question is, how do you extract a particular subregion, such as this LRR domain shown on the right? And it's a non-trivial computational problem, which traditionally has been accomplished by training models for protein domains to recognize certain sequence motifs associated with that domain. And here's a kind of visual representation of what a sequence motif is. It's basically a probabilistic model for a sequence of amino acids. And typically the way that we capture these sequence motifs is using hidden Markov models, which are trained on a library of protein domain example sequences. But as with any supervised model, HMMs can be prone to biases and fail to correctly annotate proteins which diverge from your training set. And LRRs are particularly susceptible to annotation errors because they're under immense evolutionary pressure to diversify in order to generate new binding specificities, in order to recognize new invaders. So shown here is an example where the HMM annotation has failed to capture the full extent of the LRR domain because it's truncated nearly 100 amino acids from the tail end of the LRR domain. This is the actual LRR domain. So with that background out of the way, I'll present to you our kind of proposed paradigm for protein domain annotation, which directly examines the structure of the protein obtained by AlphaFold. And we can think of this structure as a curve embedded in three-dimensional space. The centerpiece algorithm here involves the projection of the protein structure onto a 2D plane. The idea being that once you've made this projection, you can describe the slinky portion of the protein as being the segment of this projection which winds around a circle around the origin. And we do this process, which I call normal bundle framing, which is just a fancy way of saying that we are identifying different cross-sections of the coil with one another. So mathematically speaking, we need to pick an orthonormal frame for each normal plane, which minimizes variation between the fibers of the normal bundle. In other words, in order to make this identification between fibers, we compute a discretized version of a construction in differential geometry known as parallel transport. So I've outlined this algorithm here. Basically, you start by initializing a random pair of orthonormal vectors, and then you iterate along the entire length of the protein. At each step, you project the current orthonormal pair onto the next normal plane, and then that resulting projection may not be orthogonal, but then you find the closest orthogonal pair using singular value decomposition. So once we obtain this 2D projection, we compute the cumulative winding number on the resulting 2D curve. So that's this step. This is a very simple formula that we use. This is just the classic formula for the winding number of a curve in the plane. We have a discrete version of that. It's simply integrating the derivative of arctangent of y over x. So the result is that you get this cumulative winding number, which has been plotted here across the length of the protein. And you'll see that this plot has a linearly sloped segment here, which is flanked by these kind of noisy segments on the left and right. And the linearly sloped region is precisely the LRR domain. Why? Because if you take a slinky and collapse it down to two dimensions, you obtain a winding along a circle, but the cumulative winding number for winding along a circle is simply a straight line. So now that you have the cumulative winding number, how do you extract the linearly sloped region? We do this by running a piecewise linear regression with discontinuous breakpoints. And you can think of this function as being like a clipped ReLU function, allowing for discontinuities at the breakpoints. And the regression finds the breakpoints, which minimizes the sum of squares loss function. And you can visualize the result of this regression in this figure on the right, which has been colored with respect to the breakpoints. And you can see that the algorithm has done a pretty good job of isolating the region of the immune receptor, which is coiling. And it has even managed to truncate this short region on this end of the protein, which is not coiling. So once you've isolated the LRR domain, there's more things that you can do. One of the results in this project is that we're able to use some topological methods to construct cylindrical coordinates on the coil. We do this by constructing a nearest neighbors graph and then computing the eigenvectors of the graph Laplacian. And by some spectral graph theory magic, these leading eigenvectors give us a kind of dimensionality reduction for the coil. And if you compute arctangent of the ratio of these eigenvectors, we obtain a phase estimation, so like an angular coordinate along the domain. And this gives us a way of determining which direction certain amino acid residues are facing, and in particular, to decide whether or not certain residues are in phase. So I think I'm starting to run out of time. So I'll kind of skip through this. This is basically one of the applications that we had of the phase estimation. And yeah, another application of these eigenvectors is that we can detect kind of structural anomalies, anything that deviates from a coil that is sticking out, using this kind of sliding window L2 distance from the expected cumulative winding number. So in summary, there's a plethora of things you can do once you have the 3D structure of a protein. And we are now in an era of DALL-E and ChatGPT, and also AlphaFold, where we have protein structures that are more available than ever before, thanks to some powerful new computational tools. And there are hundreds of millions of protein sequences available, the vast majority of which have functions which are not fully understood. And this number is only continuing to grow as the cost of genomic sequencing continues to drop. So knowing this structure is an important first step in characterizing such proteins. We can't possibly run cryo-EM on all of them because it takes months to do this for a lab that's dedicated to it. And AlphaFold and other prediction algorithms are only going to get better with time, and this will enable us to solve biological problems with greater efficiency. It'll allow us to engineer proteins with desirable properties, to drug discovery and understanding how enzymes bind to their substrates, how multiple proteins interact. Yeah, before we go to questions, I'd like to make some acknowledgments. I'd like to thank the Krasileva lab at Berkeley and my PI, Ksenia, for being such a wonderfully supportive scientific community for me and for the last few years. And I'm incredibly grateful to have been a part of this fellowship for the last 4 years. It has had an overwhelmingly positive impact on my experience in grad school and really opened up a wealth of opportunity in pursuing research in computational biology and scientific computing in general. So big thank you to CSGF and the Krell Institute, and thank you all for listening.